Water is known to be an excellent solvent. Well, really, it's the solvent of all of life. So let's explore how through a case study. Aspirin is one of the most commonly used medications in the world. It's inexpensive, it's widely available, and it's something many people have taken without a second thought. But chemically, aspirin's not neutral, it's an acid. When someone takes aspirin, it doesn't stay as a solid pill for very long. It dissolves in water. And because the human body is mostly water, that means it dissolves into things like the bloodstream. At that point, aspirin becomes part of the body's chemistry. So, once dissolved, aspirin can release hydrogen ions, changing the chemical condition of the blood. The body doesn't allow those conditions to fluctuate freely. Many biological processes depend on chemical stability. So, to manage this, the body relies on chemical systems, that control how acidic or basic its fluids are. So, in this case study, we're going to explore what happens when too much of an acid enters the body and how that challenges the chemical balance of living systems. While it would take a lot of aspirin for an adult human to overdose, this is a real potential scenario for children, which is why we should always use child locks for medicine cabinets. In fact, this case study was inspired by a hypothetical case study by Terry Platt titled The Case of the Mortified Mom. So, to back out for just a minute, we're taking a look at one of water's life sustaining properties, its versatility as a solvent, which helps explain why water makes up anywhere from 60 to 70% of the human body. It's arguably the most critical molecule for life on Earth. And its ability to behave as a solvent, which we will discuss shortly, comes down to two main properties. First, it's a polar molecule, and two, it can form hydrogen bonds. So, water is incredibly important in biology because of what it can dissolve. Ions and polar molecules dissolve easily in water. That's because water itself is a polar molecule. It has slightly positive and slightly negative regions, or partial charges, that can surround and stabilize charged or polar substances. Because of this, water is called the solvent. The solvent is the substance that does the dissolving. The materials that get dissolved or mixed into water, like salts or sugars or acids, are called the solutes. Imagine you put a sugar cube into your coffee. You know that it doesn't just stay there floating on the surface. Instead, that sugar cube kind of disappears to the naked eye. It's dissolving in the water. Now, when we say something is an aqueous solution, all we mean is that water is the solvent, which is pretty much the case for, all relevant things to biology. And whatever else is present, like ions or molecules, are going to be the solute. This is why chemistry and biology always happens in water. Water creates the environment where molecules can dissolve, move, and interact. So how does water actually dissolve things? It does this by forming what are called hydration spheres. Water molecules are polar, meaning they have slightly positive and slightly negative ends, or partial charges. So when an ion or a polar molecule enters water, the water molecules reorient themselves around it. Around a positive ion, like we're seeing here with sodium, the slightly negative oxygen ends of water are going to point inward towards the ion and completely surround it. Similarly, around a negative ion, like the chloride, the slightly positive hydrogen ends are going to point inward. So the hydration sphere does three things. It stabilizes the ion or polar molecule, it pulls it away from other ions or molecules, and it keeps everything evenly dispersed in the solution. So this is why a salt like NaCl, which we're seeing here, which is held together by pretty strong ionic bonds in its solid crystal form, completely dissolves and dissociates when that salt is poured into water. Think of it as the water molecules pulling the ions away from each other, and then eventually the salt is evenly dispersed throughout the solution, and it stays that way because these dissolved particles are separated and mobile, just wrapped in water. This is why salts, acids, and then many other biologically important molecules dissolve so easily in the body. Water actively interacts with molecules to keep them in a solution. And once substances are dissolved and mobile, they can participate in chemical reactions, like influencing pH and interacting with buffer systems, which we're going to get to in just a little bit. And remember, much of the human body is basically an aqueous solution. So if you were to separate out the components of blood, for an example, you would find that about 55% of the total is just essentially water. Sweat and tears, so to round us off, blood, sweat, and tears, are other examples of aqueous solutions. If you've ever tasted a teardrop or like a bead of sweat, you remember that it's pretty salty tasting. That's because you don't just cry or sweat out pure water. Instead, it's an aqueous solution. Let's pause though, because I want to mention not everything dissolves in water. Non-polar molecules do not dissolve well in water because they don't have charges or partial charges for water to interact with. Without those attractions, water can't form those hydration spheres that we talked about. So a biologically important example is lipids, like fats and oils. Lipids are largely nonpolar, so when they're placed in water, they clump together instead of dissolving. You've probably noticed this if you try to mix like olive oil and vinegar. The oil droplets bead up. This is also why cell membranes exist. So the phospholipids, a type of lipid that make up the cell membranes, have a polar end and a nonpolar end. And these nonpolar tails are water-fearing, and so they orient in such a way that it ends up creating a barrier between the inside and the outside of the cell. So in biology, what dissolves and what doesn't dissolve in water shapes the structure of things like cells and can even control what moves through them. So coming back to our case study, we now understand why the aspirin that you take is going to dissolve into the bloodstream from the stomach. But what does it mean to say that aspirin is an acid? How would that impact the body, and how does the body combat this? So let's start by defining an acid. To understand pH, we need to start with water itself. Water molecules are usually stable, but very infrequently a water molecule dissociates, meaning it turns into ions. When this happens, one water molecule is basically splitting into the two ions, a hydrogen ion and a hydroxide ion. I like to think of it as the hydroxide kind of like stole the electron. It held onto an electron. Remember that a hydrogen is just one proton and one electron. So if you have a hydrogen ion and it loses its electron, all you have is one single proton. That's what the hydrogen ion represents. Anyways, in pure water, this dissociation happens at a really low rate, and the number of hydrogen ions and hydroxide ions stays pretty much equal. The important idea is this: pH is a measure of how many hydrogen ions are present in a solution. Because the concentration of hydrogen ions is usually really small, scientists use a logarithmic scale, the pH scale, to describe it. Each whole number change in pH actually represents a 10-fold change in hydrogen ion concentration. pH is calculated as the negative of the base 10 logarithm of hydrogen ions. I know, we're bringing in some math here. So if you remember learning logarithms, you can rewrite this equation with a little bit of math magic. So we're going to rewrite this, and it would be rewritten as the concentration of hydrogen ions equals 10 to the negative pH. Okay, so I told you that it's really infrequent for these hydrogen ions to form from pure water. Well, if you took pure water and you got a concentration of how many hydrogen ions there were based off of this natural dissociation of water, you would find that in pure water, the concentration of hydrogen ions is 1 times 10 to the negative 7th moles per liter. Similarly, the concentration of hydroxide ions in pure water is 1 times 10 to the 7th, negative 7th, sorry, moles per liter. I also want you to notice that the log of 1 times 10 to the negative 7th, once we write this out, right, that is negative 7. And we are taking the negative of this number, which makes it just a positive 7. Okay, so all that math stuff is basically just getting us at the fact that 7 is the pH of pure water. And that is why 7 lands right in the middle of our pH scale as neutral. Really important is that when we talk about pH, we're really talking about how water's natural balance of hydrogen ions is affected when substances dissolve into it. So acids are things that are going to increase hydrogen ion concentration when they're added to water. Bases decrease hydrogen ion concentration when they're added to water. And this shift in concentration of hydrogen ions is what the pH scale is actually capturing. Any acidic solution has a pH below 7. It's a little funky, but acidic solutions have a higher concentration of hydrogen ions than water. Basic solutions have a lower concentration of hydrogen ions in water, and they are at a scale above 7. Stick with me here. I'm going to go through some of the math that helps us conceptualize pH. One thing you have to think about is the difference between scientific notation and standard notation. So scientific notation is writing that the hydrogen ion concentration of pure water is 10 to the negative 7th. If we were to write out 10 to the negative 7th as the entire number with all of the zeros, that's called standard notation. So let's do that because, again, it helps us conceptualize pH. So 10 to the negative 7th is 0.00000001. So you saw what I did there was I took 1 and I moved the decimal 7 places. And I went left because it was 10 to the negative 7th, making this a really tiny number. Now let's do the same thing for a pH of 2. So at a pH of 2, my hydrogen ion concentration is 10 to the negative 2nd. This is the same as saying 1 times 10 to the negative 2nd. So to write this out in standard notation, I start with 1, and I'm going to move my decimal places 2 spots to the left because of that negative number. If I do that, I get 0.01. By the way, I'm being a little lazy here. This is all concentration, and the default is going to be molar, so moles per liter. So you can already see that compared to a pH of 7, a pH of 2 actually has a higher concentration of hydrogen ions. This is one of the most confusing things about pH. You would expect that a lower number is lower hydrogen ion concentration, but now that we've written this out from the scientific notation to standard notation, going from pH to actual concentration numbers, we can see that a lower pH is actually a higher concentration of hydrogen ions. I can't stress this enough. So now let's do this with a pH of 10, just to round this out. So we're going from acid now up to a base. At a pH of 10, I know that my hydrogen ion concentration is 10 to the negative 10th. Once again, same thing we did before. That's the same as saying 1 times 10 to the negative 10th. So I take 1, I'm going to move my decimal place 10 spots to the left. I get this really tiny number. So now, with all three in comparison, you can see that at a pH of 10, I actually have way less hydrogen ions, or a much lower concentration of hydrogen ions at a pH of 10 than I do at a pH of 2. Something to keep in mind about logarithms is that moving one place on the pH scale, so from 2 to 3 or 3 to 4, it seems like not that big of a jump, but it's actually a 10-fold increase or decrease. Let's look at some examples of acids and bases. So when HCl is added to water, it dissociates, meaning that it sort of falls apart into ions. And in doing so, it increases the hydrogen ion concentration. Because it's increasing hydrogen ion concentration, HCl is an acid, and it's hydrochloric acid. NaOH, when added to water, increases hydroxide concentration, and actually in doing so, it decreases hydrogen ion concentration. One way to visualize that is those hydroxide ions can react with any free hydrogen ions, creating water, and so we're decreasing hydrogen ion concentration. That means that sodium hydroxide is a base. By the same logic, NH3, ammonia, is also a base. Something I want to point out that's different between the first two equations and the third equation is that in the first two equations, we have an arrow going in one direction. That means that this chemical reaction only goes in one direction. When we put hydrochloric acid into water, it completely dissociates into chloride ions and hydrogen ions. And those two ions, they can't react to reform hydrochloric acid. Once it's done, it's done. On the other hand, our example of ammonia, this is a reversible reaction. So ammonia can react with water to form ammonium, NH4 plus, an ion, and hydroxide ions. At the same time, those two ions can reform ammonia and water. So ammonia here is called a weak base, versus hydrochloric acid and sodium hydroxide are strong acids and bases. So if a child were to take a ton of aspirin, they're essentially releasing this acid into their body, into their bloodstream, and the blood pH can start to drop to really dangerously low levels. Normal blood pH has to stay really tightly between 7.35 and about 7.45. Acidosis is going below 7.35. And very quickly, going below that can start to become incompatible with life, aka really deadly. Luckily, our blood pH is not just swinging wildly all over the place. We have what's called a buffer system, a series of chemical reactions that helps keep the pH within a very narrow range like this. So a buffer system is a system that helps resist changes in pH, even when small to large amounts of acids or bases are added to the system. In biological systems, this is incredibly important because many enzymes and cellular processes only work within a very narrow pH range, like we just learned about with the blood. Large swings in pH can damage proteins, disrupt chemical reactions, just kind of wreak havoc. A buffer usually works because it contains a weak acid that can donate hydrogen ions when the pH is going up. It also has a related weak base that can accept hydrogens when the pH starts to fall. An example of a buffer system is the acetate buffer. So an acetate buffer contains acetic acid, which is a weak acid, and then the acetate ion is its conjugate base. If acid is added to the solution, it's going to increase hydrogen ion concentration. And the acetate ion can actually react with those excess hydrogen ions and form acetic acid. This removes free hydrogen ions from the solution, then limiting the drop in pH. Now, if you imagine that a base is added, so there's an increase in hydroxide ions, acetic acid can react with hydroxide ions to form the acetate ion and water. This removes hydroxide ions, limiting the rise in pH. Because the buffer can respond in both directions, the pH stays relatively stable. That's why buffers are so important in chemistry and biology. They actively participate in chemical reactions to maintain balance. Blood is buffered to stay close to a pH of about 7.4. Here's the chemical reaction that buffers the blood. The blue arrows show the process of raising pH as more carbon dioxide is made. Reading the equation in that direction from left to right, we have hydrogen ions can react with bicarbonate ions to reversibly form carbonic acid. Carbonic acid can then dissociate into water and carbon dioxide. When you're reading the equation from that direction, left to right, you can imagine that when this is happening, excess hydrogen ions can get eaten up when they react with bicarbonate. So less hydrogen ions mean we would be making this solution more alkaline, or in other words, more basic. Then we've got the purple arrows indicating the reverse process. We have carbon dioxide and water, which can form carbonic acid, and then carbonic acid can dissociate into bicarbonate ions and hydrogen ions. This is the process of lowering the pH as more bicarbonate is created. Let's say you held your breath for longer than your body would let you. CO2 would start to build up in your bloodstream. If that's a jump, just remember that we breathe oxygen, we breathe out CO2, right? Well, that CO2 is getting produced in your cells somewhere. Like, let's say you're running, a bunch of CO2 is getting produced in your muscles, and they have to make their way to your lungs. How do they get there? The bloodstream. So if CO2 were to build up in the blood because you're holding your breath and you're not letting it go anywhere, this could cause the reaction to shift left, right? We're creating an imbalance. There's this excess carbon dioxide. The CO2 would start to react with water and form carbonic acid because we have excess, which would actually then give us excess carbonic acid, which is going to dissociate into hydrogen ions and bicarbonate in the bloodstream. This is going to acidify the blood. But don't worry, your own body wouldn't let you do this. You'd probably pass out before it got even close to that. So when an extra acid enters the bloodstream, say from a dose of aspirin, the buffer system soaks up some of those excess hydrogen ions, preventing a dangerous drop in pH. Thanks to buffers, biological systems stay chemically stable, even when conditions try to push them out of balance. However, if we have a scenario like a child overdosing on aspirin, right, the blood buffer system might not be enough to actually save their life. One treatment is actually an intravenous bicarbonate. It's an excellent choice because it's not only a base that can counteract the acid, it's a compound that's already naturally occurring in your blood. It's already a part of this buffer system. It's also a weak base, which, though it might sound counterintuitive, is better than trying to use a really strong acid, which would be just way too harsh on the biological system. So, there you have it, a crash course on chemistry and water, focusing on water as a solvent, pH, and buffering systems through the lens of a case study.